Good morning, everyone. I'm Jane Heffernan, and I have the honor of introducing our morning speaker, or afternoon speaker, evening speaker, wherever you are. Professor Deirdre Hollingsworth is a professor at the Big Data Institute at the University of Oxford. She's also held positions at the Zeeman Institute at the University of Warwick and at the Liverpool School of Tropical Medicine. She also has an honorary lectureship at Imperial College. Dr. Hollingsworth uses mathematical models and statistical analysis to study the evolution and transmission dynamics of infections with the aim of informing the design of more effective control interventions. She's particularly interested in neglected tropical diseases, and she also recently has impressive work on COVID-19. Good morning, Deirdre. We're very happy to have you here.
Thank you very much. It's great to be here. I'm just going to try sharing my screen. Just one second. All right, can you hear me now?
Yep.
And can you see my slide as a full screen?
Yes.
Okay, great. I think I might have just been talking to myself there for a minute. I wanted to start by saying thank you very much for the invitation. It's a real pleasure to be at SMB. I've never been in person, so it's really lovely to be here virtually. And I applaud the organisers for setting up the virtual meeting and also for their pricing strategy. I thought that was a really admirable approach, and I'm very happy to be here. So I was asked primarily to talk about neglected tropical diseases, which is what I'm going to talk about a little bit. But I am also going to mention a couple of things about COVID-19 at the end. So as always with these talks, there's just so many people to say thank you to, and it's important to get them up front. One of the joys of working in science is the range of people you get to work with. And I remember John Edmonds saying to me years ago, you should always work with people who are cleverer than you. And I've been very fortunate in that way. I've worked with some really fantastic people, and I will try and acknowledge them through the talk. But this is just some of the people who have been really fun to work with and delivered some really interesting science. So for those of you who don't know what neglected tropical diseases are, they're a group of diseases which are brought together not so much for their epidemiology as for the fact that they predominantly affect low-income populations. If you have good sanitation and healthcare, many of these diseases are not a problem. And often many of them could be managed through fairly simple interventions. Got some examples here. River blindness. If those of you who've been to the World Health Organization in Geneva, they have a statue related to this type of photograph here, where these people are living in an area where there's a high level of transmission of a worm which causes blindness, and the worm is transmitted by flies which live near the river. And you see that there's so much exposure to this infection that when you're a child you don't go blind, but as you get older, and then the older generations still, there was a very high probability of going blind, and you see the younger generations leading the older generations who are, and it was part of the culture. And this can be treated through repeat administration of a very safe drug. And it's just one example of diseases for which these intervention programs have had a huge impact. they are marked out in the new sustainable, well, not that new, but in the Sustainable Development Goals as a marker for improving quality of care for these populations. In 2012, the WHO and other partners led the roadmap for neglected tropical diseases, and the London Declaration in 2012 was that all of these companies and many partners were going to work together to eliminate or control 10 neglected tropical diseases by 2020. And it's really striking, the partnerships that are here. There's lots of funding bodies, lots of national governments, and it's really because people get behind this idea of really affecting the health of the very lowest income populations. And a lot of the evidence base around these interventions had been built up over several years, but they weren't that well funded. And so the Bill and Melinda Gates Foundation, along with other funders, decided that they needed to invest a little bit more in the science around these interventions as the challenges started to arise over the following years. And we were asked to form the Neglected Tropical Disease Modelling Consortium. And the Modelling Consortium is based on the idea, which was really started with Tim Hallett and the HIV Modelling Consortium, where you want multiple modelling groups looking at the same questions to try and provide robust scientific insights. And I think now in our field, this is completely standard that we try and use multiple modelling approaches. Saskia Den Boon at the London School of Hygiene and Tropical Medicine led a meeting a few years ago and wrote it up, trying to provide protocols for multi-model comparisons, which I thought was quite interesting. But we're now used to this, but a few years ago it was still slightly unusual to have groups who competed with each other to work together towards the same goal. And it's been particularly striking, I would say, in our field, in the area of onchocerciasis, that disease that I was describing earlier, where the models really were giving totally different results, and the policymakers were finding that very challenging. And those groups have really worked very hard together to maintain their scientific disagreements, but try and understand why their models are different and where they could find some evidence to try and solve those differences. And it's been a really impressive effort. So as you can see, there's lots of different institutions in our consortium, and really my acknowledgement slide at the beginning should have been even bigger than it was. So thank you to those people. Most neglected tropical diseases, and this is not all, have sort of two main areas where treatment occurs, I suppose, or sort of main methods of control. They're often divided into these two groups, but there's vector control and other aspects to it. But if we think about these two groups, mass drug administration. So mass drug administration is where you pull together a population and treat them, or you go house to house and treat them. Usually you do mass drug administration because you don't have a very good diagnostic, so you don't know who's infected, or if you only treated those who were infected, you probably wouldn't be catching all the infected. Or if you treated all those people you could diagnose, and certainly if you only treated those who had symptoms, you wouldn't be treating enough people to have an impact. You have a cheap, safe drug, and the safety profile of these drugs is really impressive. Billions of people have been treated with some of these drugs. And also it's cheap because many of them are donated by the drug companies. And you aim to get rid of infection by treating everybody in the population every year or every couple of years. And then the other type of disease, which I'll talk about a bit more later, we sometimes called intensified disease management, but it's basically case finding. So this is where you want to find the cases and prevent them transmitting onwards, but also you want to try and find the people they've already transmitted. Many of these diseases have a long time between infection and symptoms. So I often find when I give this talk that people are surprised to see leprosy on the list. Leprosy is still an infection which affects, well, I think there's hundreds of thousands of diagnoses every year, and is still a challenge in terms of reducing transmission. So as I mentioned, I'm going to focus a little bit more on mass drug administration because a lot of my research is in this area. So if we think about mass drug administration, you start off with a certain level of infection in the community and you treat, so you try and treat as much of the population as you can, and the level of infection in the population drops. it doesn't drop to 0 because you're unlikely to treat everybody. And also the drug is, of course, not 100% effective. And then we have some period of bounce back, transmission increases in the population, and then you come back, say, a year later and you treat again, and the level of infection drops, it increases, it drops, it increases, drops. And the hope is, of course, that you get down to a low enough level that it falls away. And that really introduces the concept of the breakpoint, which I'm going to talk about more in a minute. But there's also a good chance that if you stop treatment, that gradually the level of infection will bounce back. Now, for many neglected tropical diseases, this rate of bounce back is much, much slower than it is for something like coronavirus, but also for malaria, for example, is a lot faster bounce back. We're talking about much slower rates of bounce back for most of these diseases. Some of them are years, but some of them are less than that. And then if you lengthen the time between treatments, so there's proposed in some of the guidelines for some of these diseases that once you get down to this low level, you start to lengthen the interval between treatments. Well, what happens then is you sort of maybe at a new equilibrium, or you're very gradually increasing the level of infection in the population. And the sorts of questions that we address in our modelling is who should be treated, how often should they be treated, how many years is this programme going to have to go on. We talk about who gets treated, so we talk about systematic non-access. It's also been called systematic non-adherence and systematic non-compliance sometimes. How can we combine interventions to push us from the black line to the grey line? Another challenge around all of this is that mass drug administration programs have been pretty cost effective. They're often using community drug distributors. Whereas measurement, doing surveys, taking blood samples, for example, is much more expensive and more difficult to do. So as you move into an area where you might want to think about measuring elimination, your costs of your program may not be that high, but the cost of measuring 0 is, of course, huge. So there are those sorts of trade-offs being discussed now for these sorts of programs. How do you design a good survey that allows you to measure enough? And of course, there's a lot of spatial heterogeneity in these diseases, and we're now beginning to grapple with what that means as a challenge for the control of these diseases. One of the difficult things for these programs are different types of heterogeneity. And this is true for any disease that lots of us work on. I'm sure many of you think about particular heterogeneities, and I'm going to talk about a few of them now. So this is a really nice study by researchers in Ethiopia and in the USA, where they did a trial where they identified communities and they used the same intervention in all of the communities. And the black line is the average, and I hope you can see the lighter grey lines, and you see the heterogeneity in response between villages. And this can be a real challenge for these programs, that we see some villages respond well one time but not the next time. And some of that is to do with individual heterogeneity, so the fact that some people get treated in one round and not the other, some people are more infectious than others, sometimes it's that the rate of transmission happens to be higher at that particular point in time. But it does mean that you need to, with limited, this is a trial where they were really able to measure a lot. They've measured every year here. In reality, for most programs, they're measuring every 2 or 3 years, and it's quite a challenge to interpret that data. And how are we going to know if we eliminate if we can't sample every village? If we only sampled a couple of these villages, we'd get a very different story than the others. We also have huge amounts of heterogeneity between people. And for the worm infections, we have a way of measuring this. It's quite different from microparasite infections like bacteria and viruses. We actually can count how many worms people have. This is an example of a disease called lymphatic filariasis, which is transmitted by mosquitoes. And the worms live in your lymphatic system, and they can cause things like elephantiasis, so lymphedema, swelling of the limbs, or other quite severe sequelae. And this is, in green, is some data on how many worms different people in the population has. So it's a frequency plot. And you can see there's lots and lots of people who have no worms, fair number of people who have a lot of people who have some worms, and then you have a few people who have lots of worms. And one of the things we're always wondering is whether we can use this distribution as a way of indirectly measuring the biting risk heterogeneity. So we know that some people get bitten more than others, and is this a way of measuring it? If we just look at the worm burden, because that's relatively easy, well, it seems in these programs that it's easier to measure the worm distribution than possibly measuring the bite distribution, for practical reasons of how do you measure the bite distribution if you can't have human landing catchers. But that's a whole other talk, and there are people much better qualified than me to talk about it. But this measure is really important because it determines so much of the dynamics of the system. So we tend to, in the field of worm infections and macropsites, generally we fit a negative binomial often to these worm distributions. And this arises primarily, although not for all of the models, it's primarily comes from an idea that there's a gamma distribution of risk across the population, so biting risk, and then so each individual has their risk of being bitten, which comes from a gamma distribution, and then if you have a Poisson process on top of that of acquiring worms, then you end up with a negative binomial distribution of worms. Now there are complications because you don't tend to measure the number of worms, you tend to be able to measure the number of baby worms, offspring, transmissible stages, but that's the basic concept. And the parameter that we use to describe the negative binomial distribution is this parameter k. And so if we have a high k, confusingly, we have a low level of aggregation. So most people are the same. And if we go to this blue line, we have a low k, then we have a high level of aggregation, which means that this distribution over here is really extreme. There's only most people are down here, and then we've got just a few people out here. So what are the consequences for the dynamics? Well, if we have something in which everybody's kind of the same, you can reach much higher prevalence levels than if you have a highly aggregated system in which only a few people are getting the bites. So if we want to model the dynamics of somewhere that has, say, a prevalence in this diagram of 20%, there's quite a range of k values which could represent that. But then if I start to treat, if I push this system down, or for example, if I start intervening on the mosquitoes and I start killing mosquitoes, as I move down this line, if it's sort of an even distribution of mosquito bites, I can eliminate very quickly. But if I'm moving down this line, then it's really going to take an awful lot of effort. And I'm going to have to kill a lot more of the mosquitoes to eliminate the disease. So these types of heterogeneity are really important when we start thinking about elimination, which is something that's coming up for this disease. So we were fortunate to collaborate with somebody called Lisa Reimer at the Liverpool School of Tropical Medicine. And she had some data where she had measured different things spatially in different populations. And we did an analysis where we hoped to look at sort of spatial heterogeneity. I mentioned spatial heterogeneity before, but also this individual heterogeneity in bite rate, for example. So she had different measures in these same places. So she had vector biting rates, she had the levels of worms, effectively, microfilariae, so that's that plot that I showed before from a different population. So what was the distribution of worms in the population? But she also had antigenemia, which is a measure of the number of adult worms in the population. It's usually just a yes-no measure, but in laboratory conditions can be obviously a quantitative measure, and she had this data in multiple locations. And here is the data. Here we have the nightly bites going from blue to red, and in this area we have the level of microfilaria going from blue to red. And you can see that's the average microfilaria. You can see there's not much overlap between the places with high numbers of nightly bites. Now, obviously, that's a measure at a particular point in time. It's not a measure of the whole lifetime of everybody in that population. And then this average microfilaria, which is to do with quite a few years of exposure. So On top of that, in these situations, they had introduced bed nets. So here, long-lasting insecticide nets. And here we have the nightly bite total before and after the bed nets. So the bed nets definitely have an impact on the number of bites. And here we have the average, the distribution of microfilaria in the different villages. So there is some relationship, and they are ordered in the same way. There is some, you could tenuously say there's some relationship between bite load and microfilaria, but it's pretty weak. So that's not really giving us much hope that microfilaria is going to be a good marker for bites at the moment. In some ways not that surprising, but even if we looked at different population groups, we found the same thing. So what about if we looked spatially? What was the association between, for example, MF count and bite rate? As we saw from the data before, it's a pretty weak association. So one of the conclusions of this paper was that, you know, we're really getting different messages depending on how we measure these heterogeneities. So even in something like a worm infection where things are moving more slowly and we have more opportunities to measure, we still have challenges in terms of measuring and then interpreting heterogeneities, and that obviously then has consequences for the elimination dynamics. As I said earlier, I would mention one more important heterogeneity, which is heterogeneity in how the treatment is received. So this was some analysis with Louise Dyson at the University of Warwick, looking at how different models had modelled mass drug administration in terms of who gets, if you do multiple rounds of mass drug administration, is it random chance who gets treated from one year to the next? Or is it sort of semi-systematic in that some people are more likely than others? Or is it that, you know, there's certain groups of the population who always get treated and others who don't? And actually there's been some really nice social network analysis by Juliet Charmy, who's at the Big Data, just moved to the Big Data Institute, trying to understand what the social drivers are between of who gets treated repeatedly. And we found that the models are all treating this quite differently, and it has quite a different impact on their results. And we really need to think about it carefully when we're comparing models, but also actually there's not that much data, and even limited data would improve our knowledge of the situation. And then this is just some follow-on work from that spatial data I was just showing. We did some simulations looking before and after bed nets at how the heterogeneity in MF and the nightly bite rate changed. And then we did, sorry, the lines and dots are the data, and then the coloured background is the number of rounds in the model you would need to treat to get to the elimination threshold. And despite the fact that heterogeneity is changing through this treatment program, through the BedNet program. So the BedNet program is reducing the nightly bite rate, but it's also making this heterogeneity parameter small, which means the heterogeneity is more extreme. They're still getting into regions where they would really dramatically reduce the number of rounds needed to get to an elimination threshold. So as I mentioned earlier, there's two kind of ideas around elimination in the field of worm infections. The first one is the breakpoint, which comes from the deterministic models, and there's some great work by the late Robert May and Roy Anderson, analysing these models. So the important thing to remember is that there's sexual reproduction of these worms in the human host. So you need a male worm and a female worm in the human host to reproduce. As the average number of worms in the population falls, and here we've got a phase plane here. So this is transmission in the population, this is the equilibrium worm burden. If we've got too little transmission, then obviously we don't have, sorry, I'll go back. We don't have any transmission, and then as you would expect, as you increase transmission, we have a stable equilibrium, which increases the average number of worms in the population. But this point here, where we switch from one to the other, is actually is the breakpoint, and it's not quite the same concept as R0, less than 1. And it's really to do with, depending on how aggregated the population is, the probability that you end up with a female and a male worm in the same host. And this threshold here is thought to be at really low worm burdens. Sorry, my mouse, right, so this threshold here, the breakpoint, depending on your transmission rate, you if you can get below this number of average worms in the population, the infection dies away. And if you get above it, then it'll start to build towards the stable equilibrium. And this is really quite a different concept from R0, as you can see. I mean, if you have low enough transmission, you don't get low enough transmission rate, you don't have any worms. But this idea that if you could, through mass treatment, move the transmission rate would stay the same, so the number of vectors in the population stays the same, but if you can, through mass treatment, push the level of worms in the population low enough, you can actually, it'll die away. This is quite different from stochastic extinction. So where you've got, you know, we all know about branching process models. If you can maintain the branching process, then the epidemic is maintained, but if you've got low enough number of transmissions, you can die away and you have some probability of elimination. And what Emma Davis showed in this paper was that although this level is actually at really low levels of average worm burdens, actually often in some work by Edwin Michael suggests it's basically at unmeasurable levels that we would expect infection to die away. If we think about a stochastic process, actually it's conceivable that infection might die away at the sorts of levels which these programs are measuring. But she also showed that it was really sensitive to the parameters of the system, many of which are not very well known. We also followed this up with some work looking actually across the models, and this is just one of the examples where we use multiple models. So we have a deterministic model from Edwin Michael's group. Wilma Stokes' group has an individual-based microsimulation, and then this is our group. And we did a lot of simulations looking for the same sort of parameter sets, sampling across simulations in Africa. So we're doing some simulations of the whole of Africa. What are the different outcomes from the different models in terms of the probability of rebound when you get to a certain threshold? And this is really a live question now for programs. We're getting a lot of places have reached the threshold, which is defined by a particular survey, but it's basically 1% prevalence in the population. And we want to know what's the probability if they stop treatment at that level, what's the probability that it's going to resurge, and over what time scale. And so this is really an active conversation in this field. And the answer is that it's going to take years in many places. And so how do we maintain vigilance and surveillance in those settings? This paper is part of a collection of papers across a number of diseases across the 7 diseases, and also some interesting work on surveillance from Peter Dickel's group. So over the last few months, we have been doing some COVID modelling, but actually we've been most busy trying to understand the impact of COVID-19 related interruptions to neglected tropical disease programmes. So we have a report which has recently come out on Gates Open Research, and we have some related papers coming out as well. So the WHO issued guidance in, I think it was April, suggesting that mass drug administration programs, for example, should not be continued during the COVID pandemic, but that more instructions were to follow later on. And in the last few weeks, they have released some additional guidance around how to do risk assessments to see whether programs can be maintained. Now, they did advise the continuation, obviously, of some treatment programs, but things like mass drug administration were to be delayed. So we were asked to consider the question, how long can NTD interventions be postponed before progress to the 2030 goal is affected adversely? And which diseases basically is this going to be worse? And what can we do to mitigate this when we start up again? And if for the next few slides, if you kind of have a mental image in your head of you do a mass drug administration in March 2019, you were intending to do one in March 2020, and let's say you're not able to restart until March 2021. So we have this year's delay to this MDA program. So here we have a simulation, and this is the amount of heavy infection in a schistosomiasis model and in school-aged children. So this is where the sort of morbidity is considered to be most important. Well, not most important, but one of the key measures of a schistosomiasis control program. And imagine that we have a mass drug administration, we treat, it drops, we treat, it drops, we treat, it drops. But now we miss around. So we go from the grey line to the black line, and we have this difference in, oh, sorry. We would have reached elimination as a public health problem in, let's say, 2024, but because we've missed around, we're not going to get there until 2027. And that's, you know, for countries planning to achieve these goals, that's quite a long delay. But importantly, we'll also have an additional burden of infection. So the yellow area is that additional burden of infection. And of course, the longer the delay to the scheduled MDA, the greater the resurgence in infection and the greater the number of rounds you're going to have to do to come back on track. And in higher transmission settings, this is going to be worse because this bounce back rate is going to be worse. Now, it's important to note, you know, I'm looking at years here. I'm not looking at malaria timescales of months. These are still relatively slow infections. So they can tolerate a bit of a delay, but the suggestion from the models are that if you do a remedial strategy for, in this example, you do a high coverage and a community-wide round, so you have a much, when you come back strong, basically, then the delay is reduced and the difference in the burden is reduced. So across the mass drug administration diseases, we've been looking at what the potential mitigation strategies are, and acknowledging that for a lot of these diseases, the short-term problems are not as serious as some other infections, but these programs can't be delayed forever. And when they come back, they have to do extra efforts to mitigate the impact. But I also wanted to focus on some of the case finding diseases. So I mentioned that case finding is an important part of neglected tropical diseases for things like leprosy, but also this is an example of a disease called visceral leishmaniasis, which causes, we focus on it particularly in the Indian subcontinent, where it is called Kala Azar, and sadly causes death in the vast majority of people who are diagnosed, clinically diagnosed. So what happens if you have a successful case finding program? Well, let's say you start off with this level of, this is a true level of incidence. This is how many people you diagnose. So obviously you don't diagnose all cases. You start a really good program. You start to diagnose more cases. So this is always a challenge with case finding diseases, that you find a lot more cases once you start to improve your program. So for example, I know, for example, Donald Trump might say you're finding more cases because you're testing more cases. That is true to an extent, but the important thing is if you're doing it right, you start to have an impact on underlying transmission. And so you start to see true incidence fall, and eventually you should start to see your diagnosed incidence start to fall. So here we have a successful program that's going quite well. And then we start to get into an era due to COVID, for example, that the program cannot run as effectively. And in this assumption, we assume that transmission is the same rate. Now, for some diseases, the lockdowns will actually reduce transmission, but for visceral leishmaniasis, which we believe is to do with sandfly transmission in the home, it seems unlikely that there would be any reduction in transmission due to lockdown type behaviours. What happens is that you're no longer diagnosing people, so your diagnosed cases drop off because people aren't coming to health centres, for example. Your underlying transmission starts to increase, and eventually you'll start to see your diagnosed cases start to increase. Sorry. So this is very serious for these types of diseases. So if you're relying, the problem with these types of diseases, and it's not only visceral leishmaniasis, but also leprosy, for example, and other diseases, is that if your case finding is your measure of transmission, then you cannot really interpret that data very effectively unless you know something very precise about the amount of effort that is being put into diagnosing people. Or, for example, you know, you have some reliable measure, and this is not always reliable, or for example, the time from symptoms to diagnosis. Without an indirect measure of transmission, for example, a serological survey, etc., these types of data, which are the only data that are available usually, are really difficult to understand. And also there are some perverse incentives. So we published a collection in Gates Open Research, which is linked to the link I gave before, looking at the goals for these diseases. And there's an extensive discussion in visceral leishmaniasis about the fact that if you don't look for cases, you don't find them. So you can pass these thresholds, you can reach these targets, but not actually achieve the aim of the program. And in my experience, everyone I've ever known who works on visceral leishmaniasis, that would be a complete anathema to them. They are very anxious to find these cases. But the challenges of interpreting data, which is only through this one stream, it's really difficult. And that has been, of course, one of the challenges with COVID-19. So before I move on to COVID-19 itself, I just want to wrap up this piece on neglected tropical diseases and COVID-19. Of course, delays to programs or mass drug administrations will lead to increases of infection and morbidity, and therefore increased time and resources to reach the agreed roadmap targets. And that's an important consideration for a lot of the donors. But it is subtle. It does depend on the dynamics of particular diseases, and also the dynamics in a particular location, the duration of the delay, and how much coverage or success you can have in those programs when you come back online. And for the 7 diseases we work on, the biggest problems are schistosomiasis in high transmission areas, trachoma as well in high transmission areas, and then visceral leishmaniasis in the Indian subcontinent. So I'm going to, in the last 10 minutes, talk a little bit about COVID-19, but I'm really going to keep it brief because much of our work in this is not peer-reviewed. So COVID-19 is related to neglected tropical diseases, believe it or not, because it is a case-finding disease in many places. So the intervention is a combination of reducing transmission and case finding. And I'm going to talk through a couple of things related to this. So this is from not yet peer-reviewed publication from our group in collaboration with the London School, led by Emma Davis. And as we all know, contact tracing is actually it's a simple idea, but it's quite a complex problem in terms of there's so many different parameters along the pathway from someone reporting their symptoms, reporting their contacts correctly, those contacts being traced. And then we have this very long and uncertain duration of infectiousness, apparently quite variable. And also we have this unreliability, well, that's a bit strong. I would say there's challenges with the test, in that, you know, your probability of testing positive changes over time. There's that really nice paper by the Johns Hopkins group on that. And so there's been a lot of papers modeling contact tracing, trying to use contact data, you know, some really interesting stuff. And as I say, we've contributed that, looking at actually if you're able to, you know, if actually delays in testing and delays in giving people their results, for example, may actually be ways of keeping them in isolation longer and increasing the efficacy of your program. And the other thing that I wanted to mention around COVID-19 is in terms of this reduction in transmission and case finding. With Roy Anderson and colleagues, we've written a couple of commentaries in the Lancet. The last one just came out last week, and this one came out relatively early, and it was really based on some modelling we did in 2010 or 2011, I think it is, in PLOS Computational Biology, where we were looking at keeping an social distancing intervention in place for a limited amount of time. And I have to say, when we were doing that modelling, there was a pandemic report which said that lockdowns could be kept in place for 12 weeks. And I have to say we were sceptical that that was possible, but it seems it is possible, and people have indeed, you know, really changed their behaviour for long periods of time. But now we're faced with trying to prevent a second big resurgence and try and get that balance between allowing people to live their normal lives without the huge social and economic costs of the lockdown, but also preventing transmission. And it's really data and estimation which is going to transform our ability to detect and respond to resurgence. And so the second commentary here on the right, it was about estimating the reproduction number R. So in the UK, it's part of the reporting that the government makes is the reproduction number R, and more recently the epidemic growth rate, little r. And really there is huge uncertainties in these measures, and there's a lot of variation in how people are doing this estimation. And it's really, you know, I kind of thought this concept was fairly locked down in our field, but there's a really interesting questions as well as important policy-related questions around what the reproduction number is, when and how we can estimate it appropriately. And the groups who are working really hard in the UK providing this advice to government are discussing these things all the time, and I think there's going to be some really interesting things coming about to our field on this. And I just want to put in a word here for the Isaac Newton Institute for Mathematical Sciences in Cambridge, where there's been a programme on COVID-19 modelling, and there's lots of interesting talks and plenaries available online. And then I would advise you, I'm sure you're going to be saturated with amazing talks here at SMB, but if you have any time in the next few weeks to fill, I would advise you to go and watch some of those videos as well. Fascinating stuff. And just to note that, of course, human behaviour, as we all know, is a crucial part. And this idea of that there are particular population groups who are unlikely to access care, which is the same as I was mentioning earlier on systematic non-access for mass drug administration. We know in the UK, to our cost, sadly, that particular population groups were more likely to be exposed. The particular population groups were more likely to get seriously ill, and it's a real challenge to a public health programme to actually reach all those populations effectively and support them all. And that kind of pulls me through to my next point, which is elimination, which we discuss a lot in neglected tropical diseases. Even with their slow epidemic growth rates, elimination is really challenging to achieve. And then it's really challenging to measure if you're close or if you're there. And then it's challenging to maintain it. And the thing that I think policymakers find quite challenging when we discuss it is that we have to make clear to them that our models are least well validated at this point. Our models are best validated when there's a fair amount of transmission around or a fair amount of infection around. They really are poorly validated at low transmission levels. And heterogeneities between models become very important at this point, but also heterogeneities that we don't even know about become very important. The example here on the right is guinea worm, where guinea worm is a global program. The Carter Center, so President Carter of the United States, was one of the driving forces of that program. They've gone from millions of cases to tens of cases. It's a huge effort and an amazing program, really fascinating if you have time to read about it. It's just an amazing program. But as they got down to low levels, they discovered that there was possibly a transmission cycle in dogs that was possibly maintaining transmission in the human population. And again, a huge challenge for the program. So, you would never have detected that until you got down to low levels. And these sort of heterogeneities become very important at these low levels. And so we end up with these challenges of these unknown unknowns. What are we going to find when we get down to low levels of infection? And are we even measuring those populations? And this is going to be, as I say, one of the challenges for coronavirus. And one of the interesting things, for example, in Leicester in the UK, I'm fortunate to collaborate with some researchers, including Manish Parikh in Leicester, is that they're seeing a resurgence of infection, but they're not yet seeing a resurgence in terms of severe cases turning up in the hospital. So now that we're able to measure infection, which we weren't really at the start of the epidemic, we're now understanding, well, we're finding new questions to answer in terms of the dynamics of infection. And then I've put a little note at the end. This is also not peer-reviewed work led by Tom Crellin in my research group. Well, he's an independent fellow who sits in my research group. We're very lucky to have him. And he did some fairly sort of theoretical modelling looking at the impact of different patterns of immunity in the population on the dynamics, really, of, as it turned out, the analysis turned out really to be about the second peak, and how are we going to detect if we see drops in serology prevalence, and if that were, which of course a big if that was related to levels of immunity, what does that mean for the timing and height of that second peak? But as I say, that's yet to be peer reviewed, but I'm sure we'd be delighted if you had time to look and comment. So I realise I'm running out of time, so I'm going to stop in a second and reflect a little bit on what we've learned from working on neglected tropical diseases, and also how that relates to modelling infectious diseases more generally. So modelling is only a tool to be used as part of an effort to control an infectious disease, and we often I'm sure none of you would ever feel like this. I'm sure you're all very good communicators, but we often have policymakers come to us with a question, and a number of the modelers will go, we did that 10 years ago, we've done it. But I think there's a real communication gap always about what is the question that they're actually asking and what is the question that we've answered. Are they the same thing? And what do they want in terms of output? I mean, you could, how do you iterate that response and how do you work with them? And our work on the COVID-19 interruption has been another lesson for us in terms of how we do that communication. We need early buy-in and continued communication with stakeholders. And presenting results in accessible ways is actually a continual learning process. We did some work it was published in Lancet Infectious Diseases in the end on the triple drug for lymphatic filariasis. And we thought we'd done a great job. And then it turned out that everybody hated the box plots. Nobody liked a box plot. Everybody loved a survival curve or a cumulative distribution functions. So that was a real shock to me. I always thought plots were great. Not for every situation, but in this particular situation, we thought it was right, and it really, really wasn't. And and it's that learning and being open to listening and learning from other people. So, as ever, with every modeler is always complaining about data, but we can do a lot with phenomenological modelling. But really, our work relies on high quality, timely data, and an individual level data is so valuable to us, and we need to keep saying that over and over again. While acknowledging the challenges of anonymity, particularly in contact tracing data. And sharing data is the start of a conversation. And often it adds value and then gives us sort of a virtual feedback loop. Now, I know I'm singing to the choir, is that the expression? I'm sure many of you have learnt these lessons working with stakeholders, but I think in our field, we could do more to take that journey from the more theoretical end through to the more applied end. So, for example, the discussions around the reproduction number now, I think we can all help move that theoretical conversation into the applied and the outward-facing conversation for coronavirus. We all need to use that knowledge and take the time to communicate it better. Sorry, that's a bit of a preachy end, but that is the end of my talk, and I'm again going to put up this list of collaborators, but it's by no means complete. To acknowledge the Isaac Newton Institute's programme on coronavirus, and I am predominantly funded by the Lee Ka-shing Foundation and the Bill and Melinda Gates Foundation, who fund the NTD Modelling Consortium, but the funders had no input on this presentation. So thank you very much for your time, Jane.
Thank you very much, Deirdre. We do have some time for questions. Robert Smith, asks, is there a fear that the specter of COVID-19 will overwhelm efforts for NTDs, etc.? Example, funding gets redirected, researchers switch focus, meaning that there are further delays to elimination problems.
Yes, absolutely. It's a very good question. And I think everybody's very anxious about that. And I think that's why the modelling results, which suggests when you come back, you've got to come back hard, you can't come back weak, is part of that discussion. Is sort of an attempt to, it's being used in the conversation to say you can't let this slide. There was some initial concerns that our results were saying, well, you can manage a year or two, but yeah, absolutely in the longer term there's really a concern about the sort of political interest around neglected tropical diseases, and that's why the Sustainable Development Goals mustn't lose focus as well, because it really is the poorest populations. They're their health is a marker for how good your health system is. So yeah, I think everybody's worried about it and they're trying different ways to address it. I mean, it's never been the top of everybody's concern list, but they do have a long-term impact on these populations. So yeah, good question, and I don't have the answer, but it is important.
So we have another question from Michael Colbank. It seems like parameter inference with this model type could be difficult due to identifiability issues. Do you have an opinion on how much data might be needed to accurately estimate model parameters?
So I'm not sure which one he was talking about, but yeah, it's a real, so inference is a real challenge for the neglected tropical. Let's assume he was talking about one of the neglected tropical diseases. So we really have very limited data sets. They are neglected in more than one way. How much data? Yeah, it's something we are increasingly being asked, what study would you like to see done, rather than just doing secondary analysis of existing data. And I have to say that is a slight challenge to my technical skills. So I'm not that good at experimental design, and I'm so trained in trying to get the most out of the data that is available. It's quite a big change for me to think about designing programs. So to answer his second question, I have ideas, but I wouldn't say I'm robustly able to answer that question for every problem that I work on.
So another question by Daniel Olivenka. What is, in your opinion, the more effective investment strategy to eliminate tropical diseases, therapeutic interventions, or improvement of the economical situation of the affected pops?
Yeah, great question. And actually a question that I ask regularly. These interventions are actually relatively cheap. So the argument goes, although I actually can't point you to a good reference for it, that these people need help now, and these are relatively cheap programs, and so you should invest a fair amount of money in them, and then hope not only that the economic development will come along through other programs, but also that will allow people to help themselves, that you're breaking the cycle of poverty for these populations. And so it gives them that opportunity to also improve their economic state. But whether at a global level, we should be investing, how we direct those dollars towards just giving these people water, sanitation, infrastructure, healthcare. I am not an expert in that, and I must have a harder look for some better references. But my understanding is the cost of that is really in a different ballpark to the cost of the neglected tropical disease programs.
Great, we have time for another question. This is by Denise Kirshner. Fantastic talk, thank you. One question, with regard to heterogeneity you spoke about, have you considered genetic heterogeneities that render people either more susceptible or resistant as a factor? Because there's evidence for it in mycobacterial diseases.
Yeah, great question again. We just don't know, basically. Yeah, of course there must be some human genetic heterogeneity factor, and we just don't have the data really. Yeah, great question, and again an important issue, and we just don't know. Can I think of any study that has looked at it? I'm not aware of any. That doesn't mean they don't exist, but I'm not aware of any human genetic studies for the diseases that I work on that would give enough information for it to influence my models. But when we'll talk about that heterogeneity and biting rate. So that's one way of delivering what we call predisposition. So if you rank everybody by how many worms they have, then you treat them all, and then you later and they're reinfected, and then you rank them all. The rank is similar, and you can estimate what we call a predisposition coefficient. So people are predisposed to having more worms. In the models, being or a high risk of picking worms, but that's general environment. Yeah, good question again.
So another question. What procedures did you use to validate the stochastic processes and the system dynamics?
So this is probably a question about the elimination dynamics. So part of the reason we use 3 different models for the elimination dynamics is that we have, as I said, elimination, like at low levels, there's almost no data to validate. So all of the models give very similar results. If you do a mass drug administration, because there's a reasonable amount of data. And for that particular disease, there's also a very slow bounce back rate. So really the mass drug administration is just treating a certain proportion of infections. And so they're reasonably validated at that. And once you get down to low levels, that they're not that well validated, and it's not clear that the data at the higher levels is appropriate. Well, it's a necessary part, but it's not enough to validate. And that's why we try and look at parameter uncertainty and try and look at model uncertainty. So what's it called? Structural uncertainty, but we are by no means giving, like, we're trying to characterize the uncertainty as well as we can, but I wouldn't say they were validated at these very low levels, the models.
Okay, we have I guess we have a couple more questions that we can answer. How do you expect the evolution of SARS-CoV-2 to affect the heterogeneities you described?
Oh, good question. So there is some data showing that there has been some, I've forgotten the name, there's one mutation which seems to have been at low levels and is now at higher levels globally. Yeah, how do we expect? So I'd be entirely speculating. I mean, so the theory of evolution of pathogens is that you evolve to maximize transmission, not necessarily to maximize pathogenicity. So in order to detect any change of the type that you're suggesting, we really need much better data on from who's infected. So that means much better testing, and then genetic surveillance of those who are tested. And I know in the UK, there's a consortium who are doing a lot of work on the genetics, and I think we have huge numbers of sequences now in the UK and across the globe. But whether it's well tied to the epidemiological data, I'm less clear on, and that's going to be really crucial to see whether there's changes in who's getting infected and whether that's related to the virus at all. And that's also why the Leicester situation is so interesting. So Leicester is a city in the UK that was one of the first to be locked down as a city post the lifting of the lockdown nationally. And the fact that we're now seeing lots of infections in younger people and not seeing the severe cases in the older people coming into the hospitals means that we're learning a lot about transmission, but it's a lot of open questions about whether that was what happened maybe in February before we started to see the severe cases. So yeah, we'll see.
So the last question is, how many of the 2012 London goals were achieved by 2020?
Good question. I think, so none were achieved globally, but an awful lot of them were achieved in a number of, in smaller geographies. And a lot of progress has been made towards them. But there's still a long way to go. I think, so I'm not an expert, but WHO goals are aspirational to push people towards them. And actually, I would say that a lot of them are going to be met before 2030. So that the drive of not being the country, the last one standing, seems to be an important political driver. And, we've seen a lot of progress. Countries don't want to be the last country to reach a target that all its neighbours have met. So there has been a ramping up of treatment, a ramping up of data collection, and it has achieved an awful lot. But precisely your question, none. But none globally, I would say, yeah.
Well, I'd just like to thank you this morning, this afternoon, and this evening for giving such a great talk. You've opened our minds to many things, especially about heterogeneities, thinking about that. And I'm sure that there are some more questions. So if people want to ask some more questions, I'd be happy to facilitate some communication of these questions to you, if that's okay.
Yeah, of course. And thank you so much for having me. And hello, everybody. There's lots of friends I know attending. So shame not to see you all in person, but I'm really grateful for the opportunity to attend and to speak. So thank you, Jane.
Thank you so much. Just a reminder to everybody that the next sessions are starting at 9:30. Have a good morning, afternoon, evening.